Okay, the third video we have on Zebrafish is about its genome studies, the milestone and progress, and then we'll talk about the genetic manipulation and how we're going to do all those experiments. The genome is actually done on this TU or Tübingen strain. It started in 2001 from Sanger Institute in the UK. The genome already done with this 1.410 power 9 base pair. The sequence been done. Okay, we could obtain 26,000 protein coding genes. All right, there are a higher number of species-specific genes in the genome than to human and other animal models like mouse or chicken. And some of this increased gene number is likely to be, the tetraploidization, or we call this a TSD, T-specific genome duplication. Okay, for the update, you may check the website over here on Zebrafish Genome Project. So this slide shows the reference genome sequence with 25 chromosomes. So all the way down here, you can see all these different chromosomes. The blue parts are sequenced or finished. And then there are the dark area, those are the scaffolding gaps. And then the shotgun sequence or incomplete one are marked as yellow. Alright, so basically most of the sequences been reported and done. So you may check and compare this with human. So these are the major features we have from the assembly, 1.4 of 1400 megabyte, the total clone length, and the key features are protein coding gene 26,206. Only 200 something pseudogenes, all right? And RNA genes are 4,000 something, immunogenes, T-cell receptor gene segments 56. Total transcripts up to double in this, 54,000 exons, a bit more, like up to 323,000. So it's around 6 to 7 exons per transcript or per gene. The scaffold here numbers, the scaffolding N50 base pair, these are the numbers, and much less than the total length. So yeah, there are a number of contexts, only 26,000. So I think basically the completed genome sequence could be useful. Now all this data are available on the ensemble. You may check out all those different information if you need to find a particular gene, or you may study on PubMed. You can also find a gene and compare the gene with other genes. Comparing with human, we have Zebrafish here and then human here. Core relationship 1:1, 9,000 genes are core. You can see these are the main relationship. One to many, around 3,000 genes in human, but almost more than double in Zebrafish, 1 to many genes. All right. So, in other words, you have one gene in human, but perhaps you have a few more in the Zebrafish, or many to one. Only some of them will have only one copy. Okay, and there are also many to many. All right. There are awful lot total. Zebrafish around 18,000. There are 6,000 unique genes. Unique means only found in Zebrafish or only found in human. Alright, so the coding gene, the genes coding for protein, 26,000 and only 20,000 in human. Okay, but at least from this information, we also realize that there are different what we call the isoforms in fish, in human, and in mouse. So how could we tell the difference among them? We, like for example, take EGFR, epidemic growth factor receptor, as an example. You can put these gene into EGFR in zebrafish. All right, and or this mammalian ortholog will have capitals, all in italic, and this is also in italic, but then only capital in E, and then the other are lower cases. All right, and then for protein, we start with this. This is non-italic. First one is E capital, and then the other are lower cases. These are all spelled out as human, and then mouse. All right, and these are the general such as the guanine. Okay, and the genome size of zebrafish is actually half of that of human. Human is around 3 times 10 power 9, but zebrafish is 1.7 times 10 power 9. We have sequenced up to 1.4 to 1.5 10 power 9. All right. So we have obtained enough information, so we can do genetic analysis. So what are these genetic analysis that would include two things, right? Number one, chemical induced. All right, we'll come back to this later, how to do that. But mainly there are transcriptional factors or metabolic enzyme or receptors that we would use this method. We may identify the distinguished features of the mutant. Okay, and also we could produce insertional mutants. Okay, so like this example is that the melanocyte, we could identify the gene and then screen for the insertional mutagenesis. You can see this mutant, the melanocytes cannot produce myelin. Okay, so this is how we study myelin in the pigment protein in zebrafish. Okay. Insertional mutagenesis, what we can do is to do microinjection. I have explained in previous video. This is an example of outcross that we have RFP, and then we have this T2 on C. We add this gene in, and then we add transposate mRNA. When we induce them to add them together, the transposate translated from the mRNA would add this gene into it. We also crossbreed, and this is crossbreed with wild type. We do the same thing for beta-actin gene driving the expression of GFP. So you can see this one is homozygous or heterozygous. Homozygous with this RFP expressing all the way, and this is mainly for the GF3 expression driven by beta-actin. And then you may crossbreed them together, having both features, the beta-actin driving the expression of RFP. All right. And then you can see this crossing. We usually call this a forward mutation. And then in this case here, you may even study how this recombination could occur with the left or right short internal repeats. All right, and then these features, when they snap into the genome, we may study the genotype by doing PCR, say 1 to 2. You can obtain these fragments from here, 3 or 4. And then for 1,4, you do not see the 1,4, you do not see any product. So meaning that this inside here has been removed or cut out. Or in this case here, 1,4, you can see the product here that it's been shortened. Okay. So these are the different features. We will have these transposates added in. So the transposate to add these genes in, You can see the interruptions there, and you see the 1,4 primer priming this shorter sequence. All right. So meaning that the genes being flipped around. And then these are the insertional mutants. You may use a retroviral vector. Retroviral vector, of course, is from a retrovirus with the core genes from the virus removed. What we can do is to use this retroviral vector, and they have those left and right repeats to facilitate insertion. Okay, so you may package them into ribose and then infect those eggs and embryo instead of, well, you have to do a microinjection, but then the founder cell, the founder fish would have a lot of different kind of mutants. Okay, so this is actually what we call large-scale insertional mutagenesis screening in zebrafish. After you have created all these different genes, remember I told you one of those insertional mutant collection in the websites, they have created many of these mutants carrying a marker gene from the retroviral vector, and then the marker genes could be the GFP or the other color protein, and then you may cut the tail and then do the biopsy to confirm for substance dot analysis to confirm those different founder F0 and the F1 hybrid with wild type, and then F2 and so on. Just an example of what they have created. These are the different mutants after the large-scale insertional mutagenesis screening. You may select this one with traditional, the wild type as a short tail, and this one has long tail and long fin. Okay, this is called long fins. So you may check out where are the disruption, and then piece out back the whole gene. In this case, the insertion, they did not use any color gene, they used a LEC gene as a marker gene. And then you may check this different no-neck mutant in C here, no-neck mutant. And then D, nearly normal but much smaller fish. And then E, F, G, H, those are different mutants with different melanocyte pattern. This J, they call it bleached brown. Bleached brown, bleached means there's no color. Okay, so you may study all these different mutants to understand how these different genes and what they do in zebrafish. Okay, chemical mutagenesis. We have a chemical known as ENU, or ethyl N nitrosourea. This is called ENU, or ENU, ethyl nitrosourea. You treat the chemical, you treat the sperm with a chemical that you may create mutants, and then you crossbreed. So in a few generations, F1, F2, and F3, you can obtain those homozygous and heterozygous. All right, in this case, a wild type, and then you can see this notochord shortened. We call it the tail truncated. There are different mutants with strange eyes. This is a wild type here. You can see these different mutants, or these different mutants with different blood flow or hepatocytes and so on. These are done by chemicals induced mutagenesis. You may check out this link here to obtain all those different mutants. All right. A few more slides on backward genetics, including gene knockdown, because by doing microinjection, you can inject antisense RNA produced from in vitro synthesis. Okay, this is the plasmid that will produce mRNA, because you may clone the gene into cDNA from the mRNA, and then the cDNA into a vector, so that you may flip them over, and then you may transcribe to produce antisense RNA using in vitro transcription with SP6 or T3 or T7. We call it in vitro mRNA synthesis. So after you synthesize the mRNA, you may inject in, so you inject those antisense RNA. Alternatively, you may inject the whole plasmid. So the plasmid would express under the control of this promoter known as CMV, cytomegalovirus CMV promoter, which is a strong promoter that would express at different locations the antisense RNA. I have to remind you that when you do microinjection, after you don't know, there's no guarantee. You don't know exactly when this transgene got incorporated into the cells or embryonic stem cells or the different tissues after organogenesis. So we can have this what we call mosaicism occur. Okay, and of course you may make sure that the gene would get in. So you add this transposon system. So you have to add this transposase mRNA to produce a transpose enzyme. The enzyme would incorporate the gene into the genome, and that is to make sure that very little mosaicism occur because the gene got integrated into the genome in the first place. So every time after cell division, the gene is still there. Okay. Of course, if you like to do some short experiment, you may do gene knockdown, doing what we call morpholino oligonucleotide. You just synthesize this oligonucleotide, and then you inject the morpholino that you may only study the phenotype in the embryo or embryo larvae stage, because when they grow up, it will be very difficult to do so. And since we have this transgenic technique for gene knockdown, so backpack genetics, the old day we only could do gene knockdown, and now we could do gene knockout. And same finger nuclease could be used, and that would also do RNA interference, but then the nuclease could integrate the gene into it. We see the other slides, and here is the list listing all these different techniques and the techniques being used using retroviral or different mutagenesis project, and then there's a huge amount of different gene knockout available for your investigation. So if you're interested in any genes, you may just check out this website. Here just to explain to you the different techniques involving a ZFN. Zinc finger nuclease, so zinc finger nuclease, and that would guide and link with this FOC1 endonuclease. You inject this and then cleave any gene of interest, and then of course you have the binding site for the gene, so the target gene will be cleaved. So you inject this mRNA into one cell stage, and then cleavage and repair, and you can do non-homologous end joining and then screening for gene knockout. And then from founder, you may study F1 and F2. This is another example using similar technique, but this time it's known as TALEN. TALEN stands for TALEN, a transcription activator effector nuclease, TALEN. It uses the same principle, but this time you have this left RNA and right RNA. It's easier to design for the target to be pinpointed. You have left TALEN mRNA and right TALEN RNA that will join the target gene. And this time, the target gene, we have this targeting vector known as LA and RA, the left and the right, and using sequence with GFP. So with that, you may add any GFP gene into its target. In this case, we also add a promoter known as SOX2, and then with this GFP, so the GFP with the SOX2 would be integrated into the target gene of SOX2 with the SOX2 TALEN, which is the homeobox gene, and then with this technique, you can target the vector with a GFP expression under the control of SOX2. All right, I just add the chromosome 22 that they would like to target on. All right, and by doing so, you can see those GFP expression would interrupt SOX2 gene, the open reading frame that's interrupted. Finally, of course, I have to mention CRISPR, Cas9, because with this technique now you can buy a kit, design your own guided RNA or guide RNA with the sequence of interest. So the sequence of interest would hybrid onto the target, and then the guide RNA would bring in Cas9 enzyme to do cleavage. After cleavage, your genome would repair on its own by adding new DNA, and that would cause mutation. All right. So this on the right hand side comparing ZFN, TALEN, and CRISPR, and the three techniques on how we could specifically interrupt almost any gene that you want to. Okay, so with that I stop for the video on genetic analysis, and then when we come back, we on video number 4, we talk about toxicology and environmental studies. Bye bye.